Thank you very much, Adrian and Uta, for the invitation to be here today to speak with you all about the role that mixotrophic organisms play in modulating the biological export of carbon from the surface ocean. I actually did not score an invite to the discussion last February, but I did read that white paper on an airplane, and I can attest to the fact that it is excellent plane reading, and so I'm glad to see that it's open for comment, and I'm glad that the community will be reading it. It's definitely a thrill and an honor to be able to address an audience of biogeochemists because, as some of you in this room know, I really wish that I was a biogeochemist. But as it turns out, despite having tried to become one several times over the years, I'm the sort of person who gets too distracted by the details of organism life history and all the cool features of biology, and so I wound up becoming a community ecologist instead, which is how I come to be backed by this slide that shows just a few members of the surface ocean planktonic community. We've got some cyanobacteria in there, some microplankton, up to higher trophic levels, even a black box of fish and bigger stuff in there. And looking at this image, even if we just think about a few species, as we start to think about how they interact, for example, through feeding and trophic interactions, we quickly start to build some very complex food webs and complex networks of interactions among these species. And if we're taking the perspective of an interest in carbon export, it's clear that every one of these organisms has their own unique way of handling that carbon and, of course, potentially contributing to biological export through the carbon pump. For example, some of these organisms, as we've heard earlier today, will form shells that can affect their sinking velocity. Still others are particularly tasty to higher trophic levels, which can lead to their repackaging and export in fecal material. And others, although they may, say, be lysed and dissolve in situ, will contribute to dissolved organic carbon pools, which can, of course, be moved to the deeper ocean when water masses move. Now, as a community ecologist, I look at a picture like this, and I'm mesmerized, right? I could spend all day, indeed, I hope to spend a whole career, looking at some of those intricate interactions. But I'm also a mathematical biologist, right? So I understand, like everyone in this room does, that all of that complexity makes it very challenging to answer bigger questions about ecosystem processes like the biological carbon pump and its magnitude. And so a challenge that we all face is asking how much complexity we need to build in to have an accurate approximation for these fluxes. Should we be lumping all of biology into a single box? No, probably not. Is an NPZ model, for example, sufficient to a first order approximation? And of course, this is an iterative process, right? As we learn more about the biology of these systems, and as our mathematical understanding and our computational tools improve, we continue to add in new pieces to our models to improve our understanding. What I want to do today is to argue that to increase our understanding of the magnitude of the biological carbon pump, a next key piece that we need to add in is mixotrophy. So to do that, I want to begin today by defining what I mean by a mixotroph. I want us to all be speaking with the same vocabulary. And then having done that, I want to consider the ways in which mixotrophic organisms modulate the biological pump. And when we think about controls in the biological carbon pump, we usually think about three things, right? We think about primary production, how much carbon is actually available for export. So I want to think about these mixotrophs as photosynthesizers, as primary producers contributing to that carbon base. Second, we think about how that carbon is physically moving, things like particle flux. And so I want to consider how mixotrophs themselves are particles and how their sinking flux contributes to carbon export. And finally, we think about the chemical fate of that carbon. How is it being remineralized? And so I want to consider how mixotrophs and their traits modulate those remineralization pathways. And then finally, I want to take a step back and think about when and where, both conceptually and also geographically, we should be thinking about mixotrophs as playing a key role in these planktonic communities. So let's begin by getting on the same page in our vocabulary and figuring out what exactly a mixotrophic organism is. Now, simply put, mixotrophs mix two fundamentally different kinds of metabolism. We're talking about organisms that are capable both of photosynthesis, that is, using the energy of sunlight to fix carbon for growth, and also of heterotrophy, that is, taking up existing organic matter and then respiring that and using that for growth. Now, I do want to make a distinction here that by a mixotroph in this case, I'm talking about something that's grazing, something that's eating bacteria, other protists, and so on. rather than organisms that are taking up dissolved organic material. It's quite common among unicellular plankton to be able to take up dissolved compounds, and so that's not such a useful distinction. So today, when I talk about mixotrophy, I'm talking about an organism that can both photosynthesize and graze. Now, these mixotrophs are really jacks of all trades, right? On the one hand, they can use photosynthesis, which means that they have this source of carbon when, for example, their prey are limiting. And on the other hand, they can eat things, which means that not only are they getting carbon from their prey, but they also have a source of nutrients when nutrients are limiting. Because remember, when you take up that tasty bacterium, you're getting not just the carbon contained within it, but also the nitrogen, the phosphorus, the iron, and so on. So it becomes clear when we think about these mixotrophs and where they fall in these food webs that they're going to start to complicate the flow of energy and material. For example, if we just diagram our planktonic food web very simply in an NPZ model with nutrients, phytoplankton, and zooplankton, if we think about where mixotrophic organisms fall, we find that they fall at this intermediate trophic level. Many of them are capable of taking up inorganic nutrients directly from the water column. They're, of course, capable of using sunlight for photosynthesis, but they're also capable of eating phytoplankton and themselves being eaten by zooplankton. And so you can see that they can function in many ways as a sort of short circuit in this food web, changing the ways that nutrients move and the efficiency with which they're utilized, changing the fluxes of carbon, and of course, changing the ways that that carbon is exported through the biological pump. So let's think now about specific ways in which mixotrophic organisms modulate that pump. And I want to begin by thinking about them as in their role as primary producers, setting the base of carbon available for export. So on this slide behind me, I have a few examples of the mixotrophs that we find in the surface waters of our world's oceans. And I like to put this slide up because I find these organisms strikingly beautiful. They're, to me, really the charismatic microfauna of our world's oceans. And what all of these bugs have in common, in this case, is that they are photosynthetic not by virtue of having their own machinery, their own chloroplasts, but rather by stealing that equipment from another species. So these are the organisms that we call the acquired phototrophs. They're a subset of mixotrophs. And to tell you more about them, I want to focus in here on this bug front and center, which is a ciliate called Mesodynium rubrum. It gets that rubrum species name from its reddish coloration. This is the larger cell here. And it's a ciliate, has high motility, a double band of cilia around its midsection that's kind of like this double hula skirt that it uses to swim. And as it's swimming around, it's encountering prey cells, like this cryptophyte algal cell here. And when it captures one of these prey, it pulls it into its cell, plucks out the chloroplast, and then uses it for photosynthesis. So I don't know how many of you had salad this afternoon with your lunch. Whenever I eat a piece of lettuce, I feel jealous of these guys, right? Wouldn't it be nice if we could eat some lettuce and then arrange these chloroplasts on our arms, turn green, sit outside, and get all the energy that we need for the day? So at any rate, from a biological standpoint, these things are super cool, right? And even if that's not compelling to you, they're incredibly important for a biogeochemical perspective as well. And that's because Mesodynium rubrum is a bloom former. It's actually quite famous for this ability. We find it all over the world's oceans. And it forms blooms in places like estuarine systems. Here's an image from the Columbia River estuary. And also in coastal upwelling zones. This image here on the right is a satellite image taken off the coast of Africa. And you can see some swirls of red water there that are Mesodynium rubrum cells. And when Mesodynium is blooming, it's contributing up to 90% of microplankton primary production. So we're talking about something that's making a substantial contribution to the carbon available for export. Now, we might ask ourselves, here's a cell that's working with stolen goods, right? How is it so successful? How is it capable of this bloom formation? And that's a question that I asked with Mike Newbert and Matt Johnson here in the biology department at WHOI, and we decided to answer it by developing a mathematical model for acquired phototrophy. And so this model begins, of course, with our prey cell, our little cryptophyte alga cell that's swimming around. It has a growth rate and a mortality rate. And it's sharing the water column with Mesodynium, with our consumer cell. And when there's a predation event, Mesodynium then acquires the ability to be photosynthetic and to grow using the energy of sunlight. Now, this is acquired photosynthesis. And that means that when Mesodynium grows through photosynthesis and divides, the daughter cells that are produced come out in the heterotrophic state, and then they have to feed and reacquire that photosynthetic equipment for themselves. And we have mortality terms as well for our ciliate, for mesodynium. Now, looking at this diagram of this model, you see that we have two species here sharing the water column that are both dependent upon sunlight for growth. And for simplicity's sake, when we were studying this model, we said, let's just focus on that light and how that's going to govern population dynamics. And we disregarded other things like nutrients and water column physics, and instead just focused on light. And what I'm going to show you on this slide is the model's predictions for the types of population dynamics that we see as light increases from 0 at the bottom up to higher light levels at the top. And you can see that in total darkness, when light is 0 here, both of these populations fall to 0. That's a good gut check, right? We have two photosynthetic organisms. We don't want them to be able to grow in the dark. Now, if I turn up the lights a little bit, I see an equilibrium where only the prey cell, only that phytoplankton cell is present. And that's because when we parameterized this model, we made the assumption that the phytoplankton cell is more efficient at photosynthesis than mesodynium. Not only does that make good intuitive sense, right? It's working with its own native equipment. It also holds up really well to data in the literature and to our experimental validation. So let's keep cranking up the lights. Now we have an equilibrium where both species are coexisting. But intriguingly, if I continue to increase available light levels, I see the emergence of these boom-bust cyclic dynamics, right? Which is starting to be really reminiscent of the boom-bust dynamics that we associate with things like phytoplankton blooms. Now, anyone who's done modeling always has these moments of reckoning, right? Where you're looking at the model results and they're really striking and you ask yourself, is this real? Is this model telling me something real about this system? So we decided to find out, and we went into the lab and we took mesodynium rubrum cells and their cryptophyte prey and grew them in co-culture at different light levels to see if the populations would exhibit these types of dynamics. What I'm going to show you on this slide on the left-hand side are our experimental data, the numbers that come right out of those flasks in the incubators. And then on the right-hand side, the mathematical model's predictions. And for simplicity's sake, on the y-axis here, I'm just plotting the ratio of phytoplankton prey cells to mesodynium cells so that we're only looking at one set of points at a time. Okay. So let's look first at our lowest light levels. Well, it turns out that at this point, the incubator's lights are so low that the mesodynium cells actually can't grow, but the prey cells can. And so we see an exponential increase in that ratio of prey to predator over time. At intermediate light levels, that ratio first increases, but then levels off as these populations reach their equilibrium sizes. And then at the highest light levels, we see first an increase and then a decrease in this ratio because the populations are beginning to enter this boom-bust dynamic. And there are two different point shapes on some of these plots that just indicate different biological replicates. So you can see now that we're starting to believe that our model is telling us something important about the population dynamics of these mixotrophic organisms. But can it tell us more, and more in particular about the spectrum of mixotrophy? So what do I mean by that? Well, we can think about mixotrophy as a trait that determines how much carbon you're getting from photosynthesis, right? On the one hand, you could get 100% of your carbon from phototrophy, or you could get 100% from heterotrophy. And it turns out that the ciliate mesodynium rubrum that I've been telling you so much about falls on one end of this spectrum. This is a cell that's getting up to 90%, often more, of its carbon from photosynthesis, and actually only a small fraction from heterotrophy. In contrast, other acquired phototrophs, and indeed other mixotrophs, can be much more variable and fall elsewhere in this mixotrophy spectrum, all the way down, of course, to our entirely heterotrophic microzooplankton. And so we might ask ourselves, how does where you fall on this spectrum determine your contributions to primary production, and in particular, your likelihood of forming these big bloom events? To answer that question, we can go back to our model, and we can put in a heterotrophy loop, in which we allow these predation events to go directly into growth of the heterotrophic state of our consumer. And then we can vary the fraction of predation events that go down either of these pathways as a proxy for the degree to which these organisms rely on photosynthesis over heterotrophy. Now, remember that in particular, we're interested in this phenomenon of bloom formation, right? And we know from biological oceanography that phytoplankton blooms are usually triggered by changes in the abiotic or non-living environment, right? Things like the stratification of the water column early in the season, increases in temperature, an increase in nutrient availability. Importantly for our model, also changes in light availability. So what we did was to force this model to have an annual cycle in light availability, shown here in this black line up top. And you can see that if I put only the phytoplankton prey into this model, in summer, when light availability is high, I have a peak in that prey biomass. And in winter, when light availability is low, phytoplankton biomass falls. Now let's put Mesodynium rubrum, our mixotroph, into this model. Turns out that what we see are sequential bloom events, a phytoplankton bloom followed immediately by a mixotroph bloom. Interestingly, if I put in another type of mixotroph, maybe something that gets 30% of its carbon from photosynthesis and the rest from heterotrophy, or I put in something that is entirely heterotrophic, the dynamics that I observe are strikingly different, right? Now we have an organism that's capable of curtailing the phytoplankton bloom, if you compare the y-axis scales here, but does not then bloom itself. Rather, it sort of tracks the population size of its prey. So these results are suggesting to us that understanding where mixotrophs fall on this spectrum of reliance and photosynthesis is critically important to A, figuring out whether or not they can decouple themselves and their population sizes from that of their prey, and B, then using that knowledge to understand the contributions that they're making either to primary production or to respiration, with, of course, implications for the fate of that carbon through the biological pump. So having thought about how mixotrophs set the table, if you will, for carbon export, let's now think about the role that they're playing in the movement of that carbon through the water column and through particle flux. And to do this, I want to talk to you briefly about some very nice work that was done by Ben Ward and Mick Follows. It's published earlier this year in PNAS. It's a terrific paper that I encourage you to look up. And this was a paper in which Ben and Mick took advantage of the group of models called the Darwin models from the Darwin project done up at MIT. And I know many of you in this room are quite familiar with these models, but I just want to spend a couple minutes going over how they work. And basically, what the Darwin project models do is they are based in these trait-based models for planktonic communities. And what I mean by a trait of a planktonic organism is something like cell size or nutrient affinity, or whether this organism is either photosynthetic or heterotrophic. And the real ingenuity, I think, in these models, and you'll have to forgive me, I'm a bit of a fan in part because some of the first papers came out when I was in my first year of graduate school, so I really imprinted on them. But the real elegance, I think, of this approach is that what it does is it begins by seeding the surface ocean with planktonic organisms whose traits are randomly drawn from these distributions. And then it simulates this community forward in time using our knowledge of circulation and nutrient availability and looks for the emergent patterns in these planktonic communities. And these models have been really powerful, right, for understanding planktonic biogeography. They've been able to do things like predict the abundance of very small photosynthetic cells in the equatorial zones, a la cyanobacteria. They've identified diatom-like cells in coastal upwelling zones. So they've been very, very useful over the years. But just like any model, really, some of their greatest usefulness comes from looking at where they get things wrong, where we break the model. And in particular, what Ben and Mick pointed out was that the Darwin models are often wrong, particularly for the larger planktonic size classes. So if we think about 3 classes of plankton size here on the x-axis, and then measures of abundance now on the y-axis, if you compare satellite observations of chlorophyll A, shown by this black line, to the model's predictions, shown here in blue, you can see that particularly for this microplankton larger size class, the model is underestimating what the satellite data say the amount of chlorophyll A should be. And this is also true for the coupled measurement of production. So what Ben and Mick did was say, well, gee, you know, we know that a lot of mixotrophs are actually in that microplanktonic size class. Let's add mixotrophy into this model. So now, rather than saying that this planktonic cell has to be either a phototroph or a heterotroph, let's let it vary. Let's let it mix these 2 strategies. And when they did this, shown here in red, you can see that now these models are giving a much better representation of our actual satellite measurements for chlorophyll A and for production. Not only are they improving our estimations in the microplankton size class, but the models are now also predicting overall an increase in planktonic size across a wide range of classes. So if again we look at spherical diameter on the log scale here on the x-axis, and then abundance in terms of carbon on the y-axis, if you compare the model in blue without mixotrophy to one in red with mixotrophy, you can see that now we're looking at a surface ocean that's populated with cells that are in general larger in size. And because we often use, for better or for worse, as we learned this morning, planktonic size as a first approximation, a first hint at sinking velocity, it seems likely that this shift in organism size is going to have implications for carbon export. And indeed, that's what Ben and Mick found. When they compared the model without mixotrophy here, this baseline, to their model with mixotrophy, up here, this black point, they found up to a 35% increase in the model's estimation of how much carbon is being exported from the surface ocean. And there's some variation here depending on exactly how you determine the trade-offs involved with mixotrophy. But in general, it's becoming clear that at a global scale, when we account for mixotrophy in these planktonic communities, it's really changing where we think organic carbon is going and how much of it is exiting the surface ocean. So now let's think a little bit more about the fate of that carbon and what's happening in terms of its remineralization. So if we think about this mixotrophic cell, it's got its chloroplast and its digestive vacuole here. It's got carbon contained within it, and we might ask ourselves where this carbon is going to go. What pathways will it travel down? Some of these mixotrophs will be eaten by higher trophic levels, and these zooplankton will perhaps repackage them as fecal material that's exported. Others will lyse in the water column and be remineralized in the surface waters. And still others will themselves sink, and there will be some remineralization as they're sinking. Now, which pathway this carbon goes down, of course, depends on the traits of the organism involved, right? If we think about our ciliate, this bloom-forming mesodynium species from the beginning of the talk, we're talking about a ciliate that is often quite tasty to zooplankton, to copepods and the like, which may increase its export through fecal material. We're also talking about an organism that's a really great swimmer and also a relatively fragile cell, so it's less likely to sink and more likely to contribute to dissolved organic carbon pools. If we contrast this with other mixotrophs like harmful algae, like this toxic Dinophysis species, we're talking about an organism that because of its toxin production is likely to be less palatable to higher trophic levels, which of course changes the fate of that carbon. And I would argue that we have a lot of work to do as a community, both in identifying which traits are important to understanding the fate of that carbon, and then in measuring them. And it seems important to note traits related to palatability, such as stoichiometry of these cells, their toxin production, and so on. So if nothing else over the past 20 minutes or so, I'm sure I've convinced you that the biology of mixotrophy is absolutely mesmerizing and fascinating. But we might ask ourselves, well, gee, okay, that's nice, but where do we go from here? Where and when, which ecosystem should we be studying to figure out exactly what the contributions of mixotrophs to the biological carbon pump are? And at a first blush, it's tempting to argue that they should be important everywhere, right? Shouldn't they be these universally dominant superbugs? Because I argued that they can do photosynthesis when they need carbon and they can eat when they need nutrients. And indeed, there have been some studies, like this paper out more than a decade ago now in PNAS, that suggest that in some communities, these mixotrophs are able to outcompete pure phototrophs, and they are able to simultaneously outcompete the heterotrophs. But yet the fact of the matter is that while we observe mixotrophs in every planktonic community, they are actually not so universally dominant. And the reason for that, as Kevin Flynn and Aditi Mitra caution us in some of their modeling papers, is that we have to be careful not to think of these mixotrophs as the perfect beasts that are capable of doing everything equally well. Because the fact of the matter is that mixotrophs, just like every other organism, are subject to trade-offs. Things like trade-offs in, for example, energy use. And this is the point that Ken Anderson and colleagues made in a paper last year, thinking in particular about the energetic investment that you need to make to acquire resources. So if you have a cell, it has a finite amount of energy contained within it, right? And if that cell wants to, for example, acquire inorganic nutrients, it has to invest energy in building membrane transporters that allow it to take up those nutrients. If it wants to harvest sunlight, it has to invest in sometimes quite expensive photosynthetic equipment. If it wants to be heterotrophic, it has to chase things around and then digest them. And so given this finite energy budget, organisms wind up falling somewhere on this trade-off spectrum in terms of which resource acquisition strategies they're investing in. And if we couple an understanding of those trade-offs with some basic physical laws like surface area to volume ratios, we can start to understand what types of ecosystems we may expect to be dominated by mixotrophy. And this is what Aditi Mitra did in a paper that she led out 2 years ago now in Biogeosciences. And what her work showed was that when we account for these trade-offs, we actually predict that we should see mixotroph-dominated communities in these low-nutrient, mature, or late successional ecosystems. So let's think about what's happening in spring, right? The water column is stratified. You have this initial high availability of inorganic nutrients. The first thing that happens is that some of these large phytoplankton cells, your diatoms and whatnot, come in. They're taking up these nutrients rapidly and dividing quickly, and you get a spring bloom. But over time, the availability of those inorganic nutrients falls, and suddenly there's a window for our mixotrophs, right, which can continue to obtain nutrients by eating other things and continue to photosynthesize. Now, and that actually jives really well with our understanding of mixotrophs like Mesodynium rubrum, right, because this is an organism that tends to be forming blooms that show up in late summer, late in the season. And it's impossible to talk about low nutrient ecosystems, right, without immediately thinking of our oligotrophic gyres, these ocean deserts found in the middle of the world's oceanic basins, regions where nutrients are quite low. And indeed, work in these oligotrophic gyres has suggested that mixotrophy plays a very important role in them. For example, in the Sargasso Sea, Hartman and colleagues showed for a variety of stations and years that these mixotrophs are not only dominant in terms of abundance, but also playing a major functional role in terms of their role as bacterial grazers. So here on the x-axis of this plot, they're showing protostbacterivory, so the amount of bacteria being grazed by protistan organisms. And the shaded bars here are the mixotrophs. So you can see that mixotrophs are responsible for more than 50%, sometimes as high as 80% of bacterial grazing in some of these oligotrophic gyres. And this is interesting when we think in particular about who these mixotrophic organisms are. Many of them are tiny nanoflagellates, but some of them are some large cells, things like radiolarians that are forming shells that can affect their sinking velocity, with, of course, potential impacts on the biological export of carbon from the surface ocean. And when we think about oligotrophic gyres, of course, it's impossible to not then also think about their implications from a global climatic perspective. Because we know with the Earth's changing climate that we expect these oligotrophic gyres to actually expand and also to warm. So this means not only that the geographical extent of these mixotroph-dominated ecosystems is going to increase, but also that the biology of the mixotrophs themselves will be impacted. My colleague Susanna Wilkins has been working on mixotrophy for many years, and some of her work suggests that when you warm up these mixotrophs, they actually become more heterotrophic. So if we're thinking about feedbacks in the carbon system, right, we're talking now about an organism that is being transformed from a sink of carbon dioxide through photosynthesis to a source of one that is now performing more heterotrophy. So I would argue in conclusion that it's important that we think about mixotrophs as we move forward in our understanding of the planktonic food web and the export of carbon. I've argued today that mixotrophy is of course important because we're talking about photosynthetic organisms that are setting the base of primary production. They can allow for extra carbon fixation, in particular by changing the efficiency with which nutrients are used in the surface ocean. And I think it's really important that we move forward together as a community to identify the traits of these mixotrophs that govern the fate of the carbon contained within their bodies, understanding density and other traits that are related to sinking velocity, stoichiometry and other traits that are related to palatability, and then measuring them for a broad range of these organisms will be really important to determining when and where these mixotrophs should be important. And I've also suggested that taking a first step of focusing on the oligotrophic gyres would be a valuable place to look for answers to some of these questions. Thank you so much. I have my email address here for questions that we don't get to during the discussion period later. Thank you.